please welcome Dr. Gbola Amusa, Dr. Jill Weimer, Sean Nolan, Dr. Michael Hyman, Dr. Frank Sasinowski, and Dr. James Wilson.
Hi, I'm Gbola Amusa. First of all, I'd like to thank the Cura Foundation and the Vatican for hosting this important event. Years ago, I heard a really important statistic, and it was basically that due to advances in medicine and public health initiatives associated, that of all the people in human history that have ever lived to the age of 65, more than half or even more than 2/3 are alive at this very moment. And we can all appreciate the importance of public health initiatives in medicine in addressing mortality associated with childhood, and also mortality associated with the very common diseases of advanced age. But in thinking about how we create future societal gains from public health and medical initiatives, I think it's fair to ask the question, what role does or should the treatment of rare diseases have in all that? And when I think about all the discussions I've had over the years about rare diseases with various stakeholders in healthcare, including investors, scientists, et cetera, I've noticed sometimes there are a couple misconceptions. One is that sometimes the initiatives are hopeless. The endeavors are hopeless with rare diseases. And secondly, even if there is hope, the societal impact might be minimal. So I think one of the main points that we want to address with this panel is to address those two things. In my experience personally, I think it's abundantly clear, first of all, that rare disease endeavors are in fact creating a lot of successes of late, as the panelists will discuss. And then in terms of the societal impact, I can say that a lot of times the rare disease researchers are the ones that are innovating at the regulator, causing change. I can also see that the science that they're using is extremely advanced. And history has shown that when you use advanced science, it tends to trickle out to the rest of the world and be broadly applicable. And then lastly, as I mentioned, breakthroughs are happening, which you'll see across the panel, and that's creating real hope, especially for the families that are involved. And finally, I'd just like to remind that even if a disease is rare in Rome, if it's rare here, it's not necessarily rare everywhere. And when you think about even diseases that are rare everywhere, and we put all of them together, there's over 60 million Americans and Europeans afflicted with rare diseases, and over 350 million people globally. So I think it's really important that we understand better how to treat these diseases. So to start, and to get a sense of the important progress, let's watch a video to see how researchers are tackling, and in many cases, achieving breakthroughs and helping people with these often intractable, what were previously intractable diseases. So please play the video.
They are called rare diseases. 7,000 of them are presently known to medicine. A disease is considered rare in the U.S. when it affects fewer than 200,000 people, which actually doesn't sound all that rare. Worldwide, though, rare diseases affect 300 million people. 1 in 20 people will live with a rare disease at some point in their life. 50% of rare disease patients are children. One such devastating disease is SMA, or spinal muscular atrophy, the most common genetic cause of infant death. It is sometimes called floppy baby syndrome because of the profound lack of muscle function and strength caused by the loss of spinal cord motor neurons. While the most common SMA, type 1, has a mortality approaching 90%, depending on degree of respiratory function and support, a gene replacement clinical trial is showing remarkable promise. A much rarer and in some ways more heartbreaking genetic disorder is called Batten disease. Heartbreaking because afflicted children appear perfectly normal well into childhood.
Different forms of Batten disease can start to manifest at age 2 or 5. Just picture that child that you know right now. They'll start to lose their vision. After losing their vision, they'll start to decline mentally in some ways. They'll forget your name. They'll forget their own name. They'll have seizures. They'll lose the ability to speak completely. They'll end up in a vegetative state. These diseases are universally fatal.
It's almost like a thief broke into our house, and he started stealing pieces of our son. He'd come one night and take a few words. A few months later, he took Noah's ability to walk. The thief came back a few weeks later, took his ability to eat. It's taken his ability to see, to talk to us. It's now, at this point, taken nearly everything. It is a cruel, cruel disease. No parent should have to go through this. No child, more importantly, should have to go through this. And we're determined to make it a difference in this disease.
A very different but no less devastating rare disease is often called butterfly disease. That's because the skin of these children is as delicate as the wings of a butterfly. But unlike butterflies, there is nothing beautiful about this disease. Even with daily arduous wound care, mortality rates are high with epidermolysis bullosa, or EB. One of the more promising research areas involves taking EB cells, reprogramming them into embryonic-like stem cells so they can be genetically manipulated to correct the disease-causing mutation and then grown into healthy artificial skin grafts. The good news is that we are rapidly increasing our understanding of the genetics behind rare diseases, especially pediatric ones. That will not only accelerate treatments and cures, but just as importantly, the genomic study of rare diseases will further our understanding of all diseases.
The first panelist presentation will be from Jill, please start.
Excellent. So just in a way of introduction, many of you heard from my colleague David Pierce earlier this morning when he talked about our organization, Sanford Health, as an integrative healthcare system that really blends together our research and our clinical practices. And in his talk, he presented a little bit of the genomic work that we do at Sanford. Later in the conference, you'll hear about some of our cellular therapies. But really what I hope to talk to you today about is the pediatrics and rare disease work that my lab in particular does. And so I just want to kind of go back to Victoria Jackson and the Wilkie family's presentation that we heard earlier, and some of the struggles that the families face in studying and having patients with rare diseases, and really take that information that they presented about the need to collaborate, the need to share data, the need to move faster to help their children. I think that we're getting there in that model, and I'm hoping that that's what you'll hear from the panel today. And so I just want to start off, since this session is about rare diseases, and we did hear a little bit in the video about some of these, but there are many rare diseases. So individually, they're considered rare by their frequency, in that there are less than 1 in 5,000 patients to be considered a rare disease, but collective, they are not rare. There are over 7,000 rare diseases that exist worldwide, affecting in the US over 30 million Americans. So all of you probably know one person at least with a rare disease. So the approach that we've taken at Sanford is, how can we actually take approaches that allow us to tackle many rare diseases at once? And so we know that these are all genetic, many of these are genetic, and so we can't use the exact same concept from one disease to another, but can we look at the diseases as a class or a family and come up with a systematic approach to do science faster and move towards clinical programs that will actually get to patients in their lifetime. So you heard from Tracy Van Houten in the slide before, but my lab focuses on Batten disease, and I said I would try not to get emotional during this slide, but Noah, who was in the video, passed away over a year ago, and the Van Houtens lost their second child to Batten disease just a few weeks ago. So it's a very personal, you form these connections with the families, and I think as scientists, we really do listen to the families to drive our work forward. So Batten disease is actually a family of rare diseases. They're a lysosomal storage disorder that result from mutations in as many as 14 different genes. Collectively, they have a frequency of about 1 in 100,000 live births worldwide. So they're very rare, but collectively they are actually the most common pediatric neurodegenerative disease. And what they share, even though each one results from a unique genetic mutation, is a common symptomology. So in the case of CLN6 that I'm going to tell you a little bit more about, the patients typically are born completely healthy. Usually around the age of 3 to 4, they start to have language deficits, they have motor skills, and cognitively they start to decline. So they were perfectly normal, and then they start to falter, and they continue to regress over the next couple of years, so that by the time that they're 5 to 6 years old, they're completely wheelchair bound, they can't walk, they can't talk, they start to have seizures, they go blind, and they're usually dead before their 10th birthday. So it's a horribly devastating disease, and all of the forms of Batten's disease really follow a similar path and are ultimately fatal. So what our lab has really focused on is how can we throw the kitchen sink at this family of diseases, taking a multimodal approach, knowing that even when we cross the finish line with one treatment, we're working in the area of the unknown. We don't know if this is actually going to last the lifetime of the child, so we want to be armed with the second treatment in our arsenal. And so in the Batten's disease space, we work in all of these different treatment modalities, but really when we started in the CLN6 program in September of 2015 in therapy development, we really knew that gene therapy was probably going to be our fastest horse out of the gate. And so you're going to hear from Nolan from AveXis. The program that they have developed around spinal muscular atrophy was really sort of our guiding post, our framework for what we did for developing a gene therapy. And when the team at Nationwide Children's that really were the pioneers for that SMA gene therapy trial started out, they followed this natural progression of therapy development that we are also familiar with, that we spend years, as scientists doing rigorous science, making sure that we have the best drug possible to move forward. And so that preclinical phase can take us 4 to 6 years. Then we go into the preclinical or translational testing in an animal model of that disease, which usually takes another year. So by the time we get to a gene therapy program, it's 5 to 7 years before we're actually in the clinic. And as I told you with our Batten disease kids, they're often dead before they get to a treatment. So the Van Houtens, Tracy in particular, really spearheaded an enzyme replacement program that actually went to market with BioMarin under the name Malaria about a year ago. It was the first treatment ever to hit market for any form of Batten disease, and unfortunately, even though he drove really hard and really fast, the treatment wasn't in time to save his children. So what we wanted to do with the sort of the successes that the Nationwide team had done was look at their program with Brian Kaspar's help and see what they had done. And this is sort of the timeline that they had put together for their SMA work, where we do preclinical testing of the gene therapy in our mouse models. Once you have that box checked and you're good there, you move on to your IND-enabling toxicology studies, followed by your non-human primate work, and then ultimately go into human-ready GMP drug manufacturing. So that whole process for the SMA program took them about 4.5 years before they got FDA approval and moved into a phase 1 clinical trial. Our approach with Batten's disease, and in partnership with the Kaspar-Meyer team at Nationwide, is to stack all that on top of each other. And so by doing that, we have to put a lot of faith in the fact that we know that this system's going to work, and we have history and experience in knowing that it will, to be able to do this, so that essentially we can shorten that timeline of the preclinical work down to less than a year, so that we can get into clinical trials much faster. And so I'm just going to give you a couple of examples of how this worked. So I mentioned to you that most of my work to this point has been in CLN6 Batten disease. So not to throw any additional wrenches into our research, CLN6, CLN3, and CLN8, which are the forms that we mostly work on, are transmembrane organelle-associated proteins. So meaning, unlike an enzyme or a soluble protein floating around in the cell, these proteins have to get into the cell, they have to be turned on from the gene therapy, they have to integrate multiple times across the membrane, and then get trafficked around inside the cell in order to do their job. So there was a lot of speculation in the field that using gene therapy to introduce proteins like this, genes like this, might actually be toxic. But essentially, we took the vector that AveXis has now for the SMA. We cut out the SMA, put in the CLN6 gene, expressed it in cells, and showed that it was getting to where it needed to be with very, very high efficacy. If we look at just the expression of this, really what you're looking for is those red dots. We do a single intracranial injection into our mice. So we deliver the gene therapy at 1 day post-birth into the ventricles. The reason that we do that is because when we go into the clinic, the delivery method that we use in the clinic is to inject this virus into the spinal cord and allow it to flow through the central nervous system to fill the entire brain. And so we can see that when we do that, the virus is able to fill the brain. It gets rid of the autofluorescent storage material that's a hallmark of the lysosomal storage material. It rescues the neuronal death. And really, if you look at that lifespan graph in the middle, it's really, that's sort of the most exciting part, is that when we treat our Batten disease animals with the gene therapy, we're able to restore their lifespan out to a wild-type normal control mouse. So we started this program in August of 2015. We were able to stack all those parameters on top, and we were able to open our clinical trial 8 months later at Nationwide Children's Hospital in partnership with Sanford in March of 2016. So the gene therapy program has now treated 9 out of 12 patients, and we hope to release a preliminary report on our progress soon. So we're just marching through all the other forms of Batten disease. The next one we had in line was CLN3 Batten disease, which is a little bit more prevalent. And our preclinical work has shown the exact same thing, that we get great expression of this gene therapy. We're able to correct the disease. So our plans moving forward will be that hopefully we'll get FDA approval to start this clinical trial this spring or within the next couple of months. Followed right on its tails is a gene therapy program for CLN8 that we hope to complete all the work for in the winter of 2018 or early 2019, and followed right on the heels of that, we've already started our preclinical work for a CLN1 gene therapy program. So I want to reiterate that we're doing this faster, we're doing this smarter, but we're not cutting any corners. We're just using the history and working within a disease group to be able to hopefully move towards effective treatments much faster and in the patient's lifetime.
Thanks, Jill, for that great intro. Sean, please go next.
So good afternoon, everyone. I'm Sean Nolan. I'm the CEO of AveXis. And AveXis is a gene therapy company that's focused on developing treatments for rare and life-threatening neurological conditions. And what I'm going to share with you today is data from our lead program, which is in spinal muscular atrophy. And just know we are also working on programs for ALS and for Rett syndrome as well. SMA is a devastating disease. You saw a little bit of it on the video earlier, but essentially SMA can be categorized into 4 different types of, if you will, severity. SMA type 1 is the most severe, and that's what I'm going to focus on primarily today. And it's diagnosed and categorized based on a combination of genotyping and clinical presentation. So as an example, if you look at the far left-hand column there, SMA type 1 generally has 2 copies of the backup gene SMN2. The symptom onset occurs before 6 months of age. The incidence is about 60%. And really what's key here are these children with SMA type 1 will never sit unassisted. 0 sit unassisted. And what physicians will tell you, or clinicians will tell you who see these babies, that the first time they see them and that a diagnosis is made, that's the most significant milestone a baby will have. So as an example, if the child could hold their head up or they could bring their hands to their mouth, that's the last time they're probably going to do that. And then they're going to lose the ability for that to occur. And by 20 months of age, 90% of the children are either dead or they're on a permanent respirator because they're paralyzed. In contrast, type 2, you can see that there's 3 copies. There's more copies of the backup gene, which means there's more protein involved. And these kids are generally diagnosed between 6 and 18 months of age. They can live into their 30s and 40s. About 25% pass away in their early 20s, though, and these children can never stand or walk unassisted. So just to give you a little context of the continuum. But what I'm going to do, again, is focus on the type 1 kids. I think one last thing to state here is the general concept is that view, I should say, from clinicians is that these children are cognitively normal, okay? And in our own data, that's been demonstrated as well. So essentially think of this as ALS, but in infants. It is a devastating and catastrophic disease. Now let's talk about hope. So from a biological perspective, in a normal individual, we all have SMN1, the SMN1 gene. And the blue spheres that you see on this slide represent fully functioning SMN protein. So that's where we get our protein to make sure the motor neurons stay alive and we can develop and grow in a normal fashion. We all have SMN2, which is a backup gene. We all have different numbers of backup gene. But you can see that that backup gene generally produces about 10% functional protein. In an SMA-afflicted individual, regardless of the subtype, the SMN1 gene's not there or it's not working. So what the baby has to rely upon is the number of backup genes that they have, and that will impact the severity of the disease. What we're doing at AveXis, if you look at the bottom half of the slide, is that we are taking a brand new, fully functional human SMN gene and we're delivering it into the cells of these children, thus becoming the primary producer of SMN protein. So our therapy is delivered over 1 hour by an IV in the arm, and the virus takes the gene and places it into the nucleus, and immediately protein starts to work. So gene therapy is really the optimal approach to treating this disease because it addresses the fundamental root cause by putting a new gene in. When you look at our clinical data, on the left-hand side, these represent the 15 children that were in our initial phase 1 study. The vertical bars there are natural history bars, and I'll just point you to the one on the farthest right. That's at 20 months of age. 90% of children are dead or on permanent respiratory ventilation, which you can see from our data set, which was published in the New England Journal of Medicine last fall. 100% of our kids were alive at this time point. I'm very pleased to say, as of today, 100% of our children are alive and event-free. Now, being alive is one thing, but being alive and not being able to function is something very, very different. On the right-hand side, you can look at what's called the motor function scores, and you can see that the children treated with the therapeutic dose, which is on essentially the left-hand side of that scale, had very rapid and significant increases in their motor function, which is critical. You have to get protein elevation up immediately because once a motor neuron's dead, it will never come back, okay? So that's something that is done with our therapy, is a very quick onset of the protein elevation. How does this translate, though, into what children can do? And this is probably one of the most fulfilling aspects of things, is that this looks at the milestones that have been achieved by the children that have been treated with AveXis 101. And you can tell almost everyone can bring their hand to their mouth, they've got head control, they can roll over, things that you would expect a normal baby to do. But remember, take a look at the right-hand side of the slide. At the beginning, I mentioned that no children with SMA type 1 can sit unassisted. None. Zero. And remember the video of the baby that was floppy. That's what you would expect. In this data set, at the time the slide was developed, 75% of the children can sit greater than 30 seconds by themselves. It's unprecedented. And we actually just released data at the American Academy of Neurology yesterday that indicates that now we have 11 out of 12 children that can sit unassisted. So I think, just to build on what Jill was saying, what used to be considered a science experiment with gene therapy is becoming a reality. Late last year, we had the first true gene therapy approved for a rare ocular disorder by Spark Therapeutics. Hopefully, we're on our way at AveXis to be being the second gene therapy approved and bringing this to market for children with SMA. We're in development for ALS and Rets, and we want to have other programs as well. And Jill and her colleagues are working on things as other panelists are. So just wanted to say that the investment that's been going into gene therapy, the brilliant science and the collaboration is beginning to pay dividends. And thank you very much for your support and your attention today.
Thanks, Sean. Hope indeed. That was great. Michael, please go next.
Thanks a lot, Gbola. Robin, thank you so much for the opportunity to be here. Pontifical Council, good afternoon. To carry on the theme of hope and what can be done, we'd like to just highlight a few more of the details regarding the disease neuromyelitis optica, which you heard Victoria Jackson speak about earlier. And in that light, I'd like to focus more on NMO, not so much as a rare disease, but as a rare disease to the rescue. And you'll see, I hope, what I mean by that in just a moment. For the last 100 years, NMO had been a disease that was essentially an enigma, filled with mystery, misdiagnosis, and very little hope. It was first recognized in the late 1800s as a variant of multiple sclerosis. It had no known cause. The prognosis was uncertain for the last century. It's a devastating disease. It can lead to blindness, paralysis, or worse. Unfortunately, some types of relapsing diseases such as NMO have historically been stigmatized and considered mental illnesses, which helps no one. It is believed to be a rare disease, but I'll mention some numbers in just a moment that I think you'll be surprised about. Originally thought to be 1 in a million. Over the last 50 years or so, maybe 1 in 100,000, but things have changed a lot in the last 10 years that I'll mention. There is no approved therapy at the moment, and until about 5 years ago, no clinical trials. All of that changed in 2008 when a young woman named Allie Guffey was diagnosed with NMO. You've heard the story of her parents, Victoria Jackson and Bill Guffey, founding the Guffey-Jackson Charitable Foundation. That foundation was launched with the mission to cure NMO based on a principle, and that is a rare approach to a rare disease, with several goals that were brought together. First, bring experts not just in neurology, but in disciplines well outside of neurology, to come together and see the problem in a different way collectively that you cannot see from any one perspective alone. And furthermore, to do so in a way that had a sense of urgency. Lives are on the line and the clock is ticking. We brought these individuals together and urged them to rethink everything about the disease. Yes, from the molecules and cells that might be causing the disease, but also resilience. What goes right in individuals who have very similar risks but do not suffer the disease? We put forth a revolutionary way of prioritizing, funding, and monitoring progress in research. It's not enough to simply put resources on the table and hope for the best. These resources have to be actively stewarded. And that's one of the signatures, if you will, of this foundation. And more importantly, regardless of whether a study turned out positively or negatively, information, every piece of data is part of the solution. And so one of the hallmarks of the foundation was to share information as fast as we learn it. And we did that in a way that brought partnerships of capable entities together, not just those with complementary skills, but those with complementary values and goals. And so what we have today is what some people have begun to call a modern cure machine, building on the power of rare that Victoria mentioned earlier. There has been the formation of a clinical consortium of over 30 countries. This group rewrote the book on diagnosing NMO. And of course, that helps NMO patients receive the best care sooner. But it also helps patients who have diseases that look like NMO but are not NMO. In other words, it helps everyone to know what NMO is and what it is not. The CIRCLE study is a longitudinal observational study of the natural history of the disease, because knowing the disease and the experience patients have with it is the first step to understanding its causes and its cures. We've enrolled over 1,000 NMO patients, and for a rare disease, that is something. In addition, we've enrolled a number of controls that give us the comparison of what goes wrong and what goes right. The foundation applied a hybrid model, which is quite unusual to funding. It brought a business perspective of tranching and milestones and timing, as well as an academic model to blend experts that intersect disciplines in a way that brings multiple perspectives onto the problem and solving it. This has resulted in really a transformative change in the field, and I'll show you some data in just a moment. But it has turned a field that at one point had very few publications and very little recognition into something that is now sort of a lighthouse for rare diseases and beyond. And moving into the digital era, which is one of the things that the founders, Victoria Jackson and Bill Guffey, are quite expert at, getting the information out to people in a relatable way through modern digital technology has been one feature that has greatly facilitated the success and the awareness. As you can see on the left side of this figure, what was once a disease that had almost no known aspects now has about 20 steps, immunological sort of breadcrumbs back up to the cause of the disease. And that's exciting, and on the left side of the left panel, it shows the features of the immune system cells and molecules that are in the periphery. And on the right side, in the blue, is the central nervous system where the inflammation occurs. And the red in the middle is the blood-brain barrier. The immune system has to talk with the brain and the spinal cord, and we want it to do so in a way that's friendly and not in an attack mode. So we've turned a disease that's very complicated into one that has steps. And when a disease has steps, those in turn become therapeutic targets. On the right side, you can see the quantitative change that has occurred in the field. Whereas in 2008, when the foundation began, very few papers. In 2017, over 10,000 citations of more than 4,000 papers. That's a lot of progress in one decade. So today, we can say that we know quite a bit about NMO. In fact, so much so that there have been multiple clinical trials launched. We know that NMO is a loss of T cell and B cell tolerance to at least one protein, aquaporin 4, a water channel. And when I say loss of tolerance, what I mean is an identity crisis. The immune system can't figure out friend versus foe and attacks itself. This results in inflammation in the spinal cord, optic nerves, and in the brain, another thing that was not known just a few years ago. There are now validated biomarkers working with our colleagues at the Mayo Clinic and Oxford University. These are autoantibodies to aquaporin 4 and another protein called MOG. Like many autoimmune diseases, NMO afflicts women unfairly and with a great propensity, at least 7-to-1 compared to males, and often at the prime of life when families and careers are at stake. And just a side note, we think about autoimmune diseases as being rare and alone, but in fact, if you collect them together, autoimmune diseases are as common as heart disease and twice as common as cancer. So these are important diseases. As of 2018, the data suggests that there may be up to 500,000 NMO patients in the world. That's important not only to know who might benefit from these advances, but also in a realistic way to drug companies that need patients to be enrolled. And that is exactly what happened. Through these methods and these advances, now there are 3 phase 3 clinical trials nearing the finish line and others following. So this has been a quantum leap. It's been a revolution to go from no clinical trials in the last 100 years to 3 potentially approvable clinical trials in the last 5 years. And so in some ways you could think about NMO and the Foundation's approach to solving it as a potential model for cures. And I want to think with you about this for just a moment. All autoimmune diseases suffer a loss of immune tolerance. And in fact, faulty immune tolerance is really the root of many diseases. Let's take cancer, for example. Cancers emerge because the immune system missed them, or because cancer cells fooled the immune system into thinking that they're normal. That is a defect in immune tolerance. Let's also think about the duality between treatments for autoimmune diseases and treatments for cancer. So when you treat an autoimmune disease, you suppress the immune system. The results are cancer and infection. On the other side of the coin, when you treat cancer, particularly with the new checkpoint inhibitors, the side effects are autoimmune disease and infection. You can see how NMO really reflects sort of one side of the immune system coin. And the way to solve, and in fact to cure NMO, is through re-educating the immune system, called tolerization. Whereas in cancer, we want to detolerize and remind the immune system to chase down the cancer cells. In NMO and other autoimmune disease, we want to teach the immune system to be more tolerant of self. And so hopefully those examples illustrate how curing NMO really opens doors, not only to curing autoimmune diseases, but many other conditions where immune health is at the center. And so finally, you know, it was once said that NMO is the little disease that could. And I would submit to you that NMO is the little disease that is becoming a model of solving a rare disease. And as the book Victoria mentioned, The Power of Rare, tells the story of seeing a problem collectively and revealing potential solutions that you would not see otherwise, and turning and shifting the approach from sort of sledgehammer, steroids, to molecular forceps of monoclonal antibody therapy, very targeted, and in fact, so much so that each patient then becomes their own universe of treatment, that is personalized medicine. There are treatments ongoing now in NMO where cells are removed from a patient, modified in the laboratory, returned to that patient so that their immune system now understands the difference between friend and foe. That's about as personalized as it gets, when your own immune system treats you of your own disease. And so I'll just sort of finish with the concept that immune health is at the center of many diseases. And by optimizing, not maximizing or minimizing, but optimizing immune health, we feel there is a tremendous opportunity to prevent and treat a number of diseases. And then lastly, when you think about NMO as a rare disease, it is. But if you take that metaphor, one more extension, we are all rare. If you think about our own DNA, how it deals with medicines that have been developed for the average human being, there is really no average human being when it comes to genetics, genomics, pharmacogenomics, and treating a disease that is as personal as it gets with respect to the immune system knowing what to do and reminding it that it does. So I'll stop there, and thank you again for your time.
Thank you, Michael. Look forward to seeing the blueprint getting applied to other diseases as well. Next up is Frank. Please start.
Thank you, Gbola. This afternoon, I ask you here to open your minds and hearts that I may share with you learnings from my 35 years of developing therapies for our sisters and our brothers that are affected with rare diseases. Kindly let me share 4 keys first to understanding why a focus on rare diseases helps everyone. First, rare diseases open the door to scientific breakthroughs. Second, rare diseases change the regulatory system to be more flexible. 3, therapies for rare diseases in the United States help many elsewhere in the world. And 4, persons with rare diseases are leading the revolution in drug development that is recognizing the importance of patients. So rare diseases open the door to breakthroughs. An antisense oligonucleotide, or ASO, is a single strand of nucleic acid designed to bind to the messenger RNA to turn off the defective gene causing the disease. FDA has approved 4 ASOs. I've aided on 3 of these, including the 2 most recent ones shown here. Exondys 51 was approved on a single-arm trial in only 10 boys. This challenged regulators to be flexible. When I was selected to unblind the pivotal study for those with type 1 SMA floppy babies with an ASO, I opened my data binder and I cried, because the infants on the placebo behaved just like the floppy babies that Sean showed you, while those on the ASO appeared to be nearly normal, picking their heads up, turning over, and acquiring many other motor milestones. As for gene therapy, Sean has already shared, and Jim is going to share with you about their gene therapies. All I can say to the rest of you is know that there are many, many more on the way. With respect to cell therapies, I wanted to share with you something beyond what you're going to hear in the rest of the conference. That is, you're going to hear later about stem cell therapies. I want to share with you some examples of, for instance, I have here 2 examples of cells being used in 3D printing. I met with the FDA recently on the use of 3D printing of a mix of polymers and autologous cells to form ears and someday noses. You know, most orphan diseases affect infants and children. And those that I meet, those children tell me that their deepest desire, what they pray for, is to be normal. If any of you have seen the movie Wonder with Julia Roberts and Owen Wilson, you will see that's about a boy who is born with only part of a face. Imagine the miracle someday when he can have, who was born without either ear, when he can have ears and be normal. Another project that I'm working on with 3D printing is the use of 3D printing of a collagen scaffold onto which autologous cells are grown to form a new lung or kidney that won't be rejected and won't need immunosuppressive drugs. As for live bacteria, this month I met with the FDA about an oral live bacteria to treat a very rare disease. Xenotherapies. FDA in 2009 approved the first drug made using a genetically engineered animal. It was a female goat genetically altered to produce human anti-clotting protein in its milk. And xenotransplantation may even more dramatically open the door to science. As recently I met with the FDA on a program to harvest organs from pigs genetically altered to overcome rejection when they're transplanted into humans. Rare diseases also drive regulatory innovation. Abby Myers, the mother of 2 boys with Tourette syndrome, and Marjorie Guthrie, who's the widow of the folk singer Woody Guthrie, who died of Huntington's disease, they led a patient movement in the late 70s and early 80s that resulted in the 1983 Orphan Drug Act, which jump-started the global movement to develop therapies for rare diseases. When the AIDS crisis started, it was a rare disease. AIDS patients in the mid-1980s threw blood at the FDA building. That got our attention when I was at the FDA. These patients moved us at FDA to create new ways to approve drugs. instead of requiring evidence of overt benefit, FDA declared that for certain diseases, that is, those that are serious diseases without any available therapies, a drug could be approved that merely showed that the drug was, quote, reasonably likely to predict benefit. I will show you in the next slide the power of the Tropical Disease Priority Review voucher. But as for orphan drug flexibility, in 2010, I analyzed all the drugs that FDA had approved for rare diseases up till that time. What I found, I then shared with the FDA, drug industry, academics, the investment community, and patients, and that is this, that the FDA had been remarkably flexible in these approvals in the past, and the FDA was therefore empowered to be flexible in the future in its regulation of drugs for rare diseases. This was a watershed moment and changed everything. And therapies that are rare in the United States do benefit many others elsewhere. Yesterday was World Malaria Day. The Tropical Disease Program that was created in 2007 in the United States, only 2 years after it was developed, led to the approval of Coartem, the first drug for malaria. And more recently, on August 29th, go down to the last one, on August 29th, 2017, FDA approved the first drug for Chagas. If you don't know it, it's a disease that kills more in South America than malaria. And it disproportionately affects the poor, as an insect that can live on the mud walls may bite a child, transmit a protozoan that travels to the heart and lives there, eating away at the muscle until the heart fails 20, 30, or 40 years later. The Argentine government celebrated this public health quantum leap by hosting an event at its embassy in Washington in November. See the woman who's between the embassy's consul general and me? That's Dr. Sylvia Gold. Her father, a pharmacist in Buenos Aires, started Mundo Sano, or Healthy World, to attack Chagas in the only way it could back then, by vector control. It's appropriate that a patient advocate is at the center of this photo because patients are always at the heart of our passion for seeking cures. Here is a photo there in the large group of one of my colleagues with patients who have a more mild form of SMA. These are the ones that Sean showed you. These are types 2, 3, and 4. In the other photo is Ella Murray, a 13-year-old with epidermolysis bullosa, what you heard about in the opening video as butterfly disease. I met Ella when I asked to sit next to her at a gala dinner in New York City in October. Meeting Ella moved me. I organized a day-long meeting. I'm sorry, I'm like Jill. I organized a day-long meeting with Ella and 60 other EB patients and senior FDA officials just 20 days ago. I can still picture them, April 6th. At the end of that day-long meeting, FDA officials spoke of how much these patient testimonies affected how FDA will regulate EB therapies in the future. So, from Abby Myers and Margie Guthrie marching in Washington with patient advocates to create the orphan drug movement, to the AIDS activists who pushed us at FDA to do more, faster, to all the patient advocates around the globe, like Ella. The rising power of the patient voice is not to be denied because it stems from love. It speaks of love. It touches the love that is God in each of us. And we move forward together in love as one. Thank you.
Thanks, Frank. The regulator is clearly a gating factor for breakthrough innovation, so thanks for the work done there. Lastly, and not least, is Jim Wilson, one of the great innovators in gene therapy behind some of these breakthroughs. Please, Jim.
So as we heard from Jill and Sean earlier, gene therapy is a pretty simple concept. Correct a gene in patient cells in which those patients do not have that gene. Unfortunately, the simplicity of the concept really belies the challenges of its successful execution. I've tried for 35 years from a technical standpoint to make this work. The technical goal of gene therapy is to inject a gene, have it target the desired cell, whatever that may be, a motor neuron for SMA, and deposit the gene in the nucleus, and all the while evading your immune system that's going to try to reject it. If accomplished successfully, as we've seen with SMA, this could potentially lead to a permanent residence of that gene in our cells and potentially a long treatment, if not a cure. So being the scientist, I wanted to bring a layer of science to this, which is the eureka moment in the 1980s that made this whole concept a reality. And that was the notion of using a virus to deliver normal genes. The challenge is to eviscerate the virus of its pathogenic genes and incorporate into the virus the therapeutic genes, thereby creating a stealth-like gene delivery vehicle. This is a picture of a virus. It's not that large, it's smaller, amplified up, and it's called an adeno-associated virus, or AAV, and my lab discovered this as a human pathogen at the turn of the century. From the discovery of these viruses, we engineered them so they would have the properties that we would hope that they would have, and they're called AAV vectors that are used in many of these studies. So I want to share with you just two examples of this new platform of AAVs that have been used in addition to the incredible story that came out of Nationwide Children's for SMA. So the first is to treat a disease called hemophilia. Hemophilia is a rather common rare disease due to genetic defects that affect boys that lead to a decrease in production of a protein that's necessary to clot. In developed countries, there is a standard of care, which is a protein infusion. And these boys, for their entire life, receive an intravenous infusion, as you can see by this boy, 3 times a week, thereby successfully replenishing the level of protein in their blood. The problem is proteins don't last forever. So there is a burden of treatment and a burden of cost. So the cost of these therapies is approximately $300,000 a year for the life of the patients. Nevertheless, can lead a relatively normal life. So how do we envision gene therapy for hemophilia? We would take the gene encoding the protein that's made in the incubators to inject the patients, clone that into an AAV vector, and then inject that vector into the blood, target the liver cells, which is where it's produced, and reprogram those cells to make the factor IX protein for this form of hemophilia. So this is the concept. You see the arrow, that's a one-time infusion, and the level of serum levels of factor IX that do not dip up and down, but they're perfectly stable because we permanently engrafted those cells with a gene, the so-called one and done. The work that was initially done, as Jill had highlighted, really needs to be done in preclinical studies, and in this case, dogs that had an inherited form of hemophilia, just like humans, were treated. And in this work done in my lab, shown in this panel, a dog that was born with hemophilia received an infusion of these AAV vectors, ultimately the one that we think works better, AAV8, and you can see the blood levels spiked up, and they stayed there for 12 years. The dog died of normal causes. This then was translated into human studies and pivotal studies in 2012 in patients with hemophilia B in a paper published in the New England Journal. Some of these patients who received an initial injection of vector are still without the need for protein replacement 9 years out. So I want to end with a wonderful story of a young boy named Corey Haas, who was born with an inherited form of blindness called Leber's congenital amaurosis type 2. His story was the topic of a popular book, and a colleague of mine at the University of Pennsylvania, Jean Bennett, dedicated her life to the treatment of these diseases. Using an AAV vector expressing a normal version of the gene that's defective in Corey, the team attempted to treat his blindness by injecting the vector in the back of the retina. The results were really nothing short of miraculous, and with the luck of some technology. This is a movie of Corey before he received treatment, trying to navigate an obstacle course with Gene. So he couldn't see that block. Gene, nervous in the back. Corey says this is hard. Is clearly anxious about being able to successfully navigate this. Again, Corey tells Gene this is hard, indicating he can't see anything. And eventually, so after gene therapy, and these were not time adjusted, this is Corey again, obstacle course. Corey can now play catch with his friends. This is a work of Gene Bennett. Quite a character.
Yeah, the door should be bigger, you're right. I think it should be.
So what's the future? That work, again, came out of an academic lab, formed the basis of the first approved gene therapy product by the FDA just a few months ago, as I commented. I do want to point out, though, that we're only at the beginning of a revolution here. Our work is not done. And in fact, the diseases that I've highlighted, hemophilia, LCA2, and what Sean described, SMA1, are the easiest. That doesn't necessarily mean they're going to translate to every disease. But the lessons that we're learning and the technology that we're developing will allow us to rapidly accelerate. And I would argue, too, that we need continued investment and innovation. And just one final comment is the sort of the next wave of innovation, genome editing, that we're going to hear about much more tomorrow. And this is the next level of gene therapy, where technology designed to not only replace a normal gene, but to go in and to fix mutations. We look forward to that session tomorrow as well. Thank you.
Great. I'd like to thank the audience. Special thanks as well to Jill, Sean, Michael, Frank, and Jim for not only creating new hope, but also on delivering on the hope that was generated in years past. Thanks, everyone.